Mokola virus is a RNA virus related to rabies virus that has been sporadically isolated from mammals across sub-Saharan Africa. The majority of isolates have come from domestic cats exhibiting symptoms characteristically associated to rabies virus infection. There have been 2 reported cases of humans being infected with Mokola virus. Classification: Mokola virus is a member of the lyssavirus genus, which belongs to the family Rabdiviridae. MOKV is one of 4 lyssaviruses found in Africa. The other 3 viruses of the classic rabies virus, Duvenhager virus and Lagos bat virus. Genome and structure: the MOKV genome comprises a non-segmented, single-stranded, negative-sense RNA molecule of 11,939 nucleotides in length. It is estimated that 90.3% of the MOKV genome consists of protein encoding a signal encoding sequences. The genome encodes 5 proteins: matrix protein M, transmembrane glycoprotein G, nucleoprotein N, phosphoprotein P, and the RNA-dependent RNA polymerase L. MOKV is an enveloped virus, with the envelope lined with spikes consisting of the viral protein G. MOKV exhibits the bullet-shaped morphology that is characteristic of members of the Rabdiviridae family. Evolution: this virus appears to have evolved between 279 and 2034 years before the present. Transmission: the human cases of human MOKV infection have demonstrated the zoonotic potential of MOKV, but as a natural reservoir has yet to be identified. Transmission of the virus is poorly understood, considering the MOKV isolates from small mammals and domestic cats. It has been suggested that small mammals, which are preyed upon by domestic cats, may be the natural MOKV reservoir. Unlike other lyssaviruses, MOKV is able to replicate inside mosquito cells in vitro, suggesting that insects may be implicated in MOKV transmission. Pathology: symptoms provoked by MOKV infection are very similar to the symptoms associated with rabies virus infection, such as fever and headaches, for example. Ultimately, both MOKV and rabies virus infection can lead to death by encephalitis. The similarity between the symptoms associated with MOKV and rabies virus may be the reason why there have been so few cases of recorded MOKV infections. MOKV infections may be being falsely diagnosed as rabies virus infections. There is one significant difference between the symptoms observed in domestic cats infected with MOKV and rabies virus. MOKV-infected domestic cats do not demonstrate the aggressive, unprovoked behavior traits typically associated with rabies virus-infected mammals. Control: at present there is no medical or veterinary vaccine that protects against MOKV infection. It has long been established that in spite of the relatedness between MOKV and rabies virus, immunization with the rabies vaccine does not confer protection to MOKV infection. Isolation of MOKV from a number of domestic cats vaccinated with the rabies vaccine have demonstrated this fact. In order to neutralize rabies virus, the rabies vaccine targets the transmembrane glycoprotein G on the viral envelope. Variation in the amino acid sequences of the antigenic domain 3 within protein G between rabies virus and MOKV has rendered the rabies vaccine ineffective against MOKV infection.